Greetings and welcome to our next installment of our video series. This is 3.3, Tissues, Histology, and Pathology. In this video, we're going to look at what histology is, what its connection is to pathology, what are tissues, where do tissues come from, and in what ways are tissues different. Before we get into that discussion, I want you to take a moment and look at this postcard. This postcard is not a mistake to include in this video, but before you would guess where this postcard came from, you would probably notice some things. Perhaps you noticed the three ladies there in grass skirts. Perhaps you noticed the sandy beach, the ocean behind them, the palm trees. And if you had to guess where this postcard was from, you would say what? Fuquay. No, you probably would not say Fuquay. You'd probably say Hawaii. Well, how did you know? Because nowhere written on this postcard does it say Hawaii. You were able to look at all the pieces and infer, sort of piece together where this might have come from based upon all the elements. In a similar way, when you look at tissues, you need to look at the small details, look at the pieces. As you really begin to look at anatomy and physiology, especially at this level, the histological or tissue level, you have to pay attention to the details. Perhaps you may recognize what this tissue is, but even if you didn't, you might notice um, the large cells that are very prominent here. You may notice these cells here and there and there. You may notice these extensions that come from them. You may notice also some small cells that are sort of in between all through here. And if it wasn't clear, maybe you could zoom in here and see how this is all kind of fuzzy areas as well, indicating more connections. So perhaps you know now what we're talking about here. This represents nervous tissue. But again, just as we saw before in our dancing ladies at the beach in Hawaii, we didn't see the name there, but we could piece it together. The point of this is when you are in lab or you're learning tissue types, pay attention to the details, zoom in, because even at this level, anatomy reflects physiology. So what is a tissue? A tissue is going to be a collection of cells or a group of cells with a common purpose. And we will look at those purposes a little bit later in these video series. But here you see the four examples of tissues. You notice that we have we'll start over here with our organ. Obviously, our organ here is the stomach, but notice the stomach is made up of tissues. We see muscle tissue, connective tissue, nervous tissue, and epithelial tissue. These are the four major tissue types. They're also showing you blood. Blood is actually an example of connective tissue. Histology, then, is going to be the study of tissues. So why do I include a slide here about pathology? What's the connection between histology and pathology? The connection between the study of tissues and looking at pathology, which you should know is the study of disease. So a histologist may be, we'll start with a pathologist. A pathologist may be a person who is going to be looking at tissues to find abnormalities. One way that that tissue might be obtained, you see an example here of a needle biopsy. In a needle biopsy, some fluid is removed from a suspected pathologic area, a disease area, and then perhaps this pathologist is going to look at that tissue, look at the cells derived from that biopsy, and look for abnormalities. Knowing what normal tissue is like from histological studies could help the pathologist determine if this tissue is abnormal, and if it's abnormal, in what way is it abnormal. It might indicate an early stage of disease or a later stage of the disease. Where do the tissues come from? When we're talking about the tissues, the four main tissue types, and once again, they're muscle, nervous, connective, and epithelial. Where do they come from? These actually arise early after a zygote is formed. A zygote formed by the sperm and the egg will go through a series of cell divisions, and you see a blastocyst, which is a stage of early embryonic growth. We get to the gastrula stage, and then we start seeing these tissue layers called germ layers form. You see this term's a little hard to read here: the mesoderm, endoderm, and ectoderm. Why we show this is that even at this very early stage, when you don't have anything, you don't have eyes and a heart, anything of that organ nature, you form these early tissue layers. Notice that the ectoderm, those layers, those particular cell types will eventually form parts of the epidermis, neurons of the brain, pigment cells. The mesoderm will eventually give rise to cardiac muscle, skeletal muscle, etc. So early after we have a zygote formed, and early after cell division occurs in early mitosis, we get this process called differentiation. That is, cells get locked into pathways. And even at this early stage, this is going to be about uh, a couple of weeks after, well, 7 to 8 days after we have fertilization, we're going to start seeing some pronounced tissue formation here, and this is going to give rise to ectoderm, mesoderm, and endoderm. And again, these are called germ layers. Germ not for disease, but for germinal, for to produce. So early on, even before a woman is pregnant, these early divisions are occurring to produce these tissue layers that will eventually give rise to our 4 major tissue types. Why is it important that we know this? Well, when you think about what happens early in development, you can see some of the effects if the tissue has abnormalities. For example, if a woman is drinking during pregnancy, alcohol consumption may lead to a condition called fetal alcohol syndrome, or FAS. Um, it seems kind of hard to believe, but 3 to 6 weeks after conception, those layers are forming. And if there's mistakes or there's environmental toxins, as alcohol would be, around those early embryonic layers, then a condition like fetal alcohol syndrome may emerge. In this case, alcohol damages particularly nervous tissue, so that children are usually born with FAS who have cognitive abnormalities. They tend to have issues related to um, ability to, oops, ability to think. They may have some behavioral disorders because alcohol damaged the nervous tissue way back, even at 3 to 6 weeks. So why are different tissue types at all, and how do they differ? We'll look at each of the 4 tissue types briefly in subsequent videos, and we'll talk about one in class. But they differ in several ways. Some of the tissues are going to have a very fast and quick rate of cell division, and some really don't divide at all once they are formed. Some are going to have very close connections, very tight connections between the cells that make them up. Some are going to have very loose connections or even no connections. They will also differ in vascularity. Vascularity means how much blood is delivered to them, how vascular they are. As you'll see, there's going to be quite a difference there as well. And even primary functions. You will be responsible for knowing the primary functions of each of these 4 tissue types. And we'll be talking about that in subsequent videos.